Yeah, I mean, just forget about this carbanion transition state and adding water across the double bond. I suppose there's a way to talk about it that way, but we're not talking about it that way. So big X. So it's simply to make the substrate specificity point. Okay. Yeah. Okay. Last step. Highly unfavorable, but pulled forward because the oxaloacetate concentration is kept very low. And is that a problem for the next reaction, which is the first one, the citrate synthase? No, because citrate synthase can be thermodynamically favored by the fact that you're creating a high energy dioester bond acetylchloride. Okay. So that is then. That was a reaction to the TCA cycle, and then we emphasized where's the energy yield coming from, these 4 reduction steps, or rather these 4 oxidation steps to generate 4 reduced compounds per turn of the cycle, which means that if you're counting from a glucose, it's 2x on all of these. Right? And an NADH is going to be worth 2.5 ATPs, so every NADH here is worth 2.5 times 2 is worth 5, so you've got 15 NADHs just out of those 3 reactions. Right, sorry, 15 ATPs out of those 3 reactions. So here's the ATP arithmetic. And adds up to, per glucose, it adds up to either 30 or 32, which depends, and the distinction depends on how these NADH electrons are used, the ones that come from glycolysis. How are they going to get used? Are they going to be able to be used in the same way as the NADH electrons are when they're generated from inside the mitochondria? Or is there going to be a problem because they're outside and the reactions occur on the inside of the inner membrane? We've got to move the reducing equivalents across. Is that going to be efficiently done every time so that we get the full power or not? And the answer is that sometimes it is and sometimes it isn't. And that depends on actually the cell type. So we'll hit that point later on. Okay. Any questions? So for now, the only thing you really need to avoid, you need to know, you kind of have to memorize the number 2.5 or 1.5 for FADH2. That's why it's 3 here, so that if you can actually just figure out how much ATP there is. But it's just a purely numerical exercise. Okay, so let's move on. And I'll talk for a little bit about some other aspects of metabolism that have to do with gluconeogenesis and the metabolism that is connected to how glycogen is formed and broken down. And, excuse me, one of the reasons that I'm skipping around a little bit in the book is so that we get all the reactions actually covered first, so that we see what they are. And then we'll talk about the regulation of them. The textbook doesn't really do that. It just gives you glycolysis, and then before you know it, you're already talking about how it goes backwards. So I just thought it was better to organize the material this way. Okay, so here we've got, so we're going to be talking next week a lot more about the fact that there's differentiation of function in different organs in the body. And so this is where we get to talk a little bit about physiology and biomedicine. But the, a key distinction is between liver and muscle. And muscle is a selfish tissue in the sense that it's doing the work. And so when it gets energy stores, like from the bloodstream in the form of glucose, it uses them right away. Or if it builds up some glycogen in its own tissue and it breaks it down, it uses it for itself. So the muscle is a selfish tissue. Liver is an altruistic, generous, I care more about you than me kind of tissue, because what liver will do is accept the sort of waste product from muscle, which is the lactate that builds up when it goes anaerobic. And then through the process of gluconeogenesis, right, we're going from the 3-carbon lactate back through pyruvate all the way up to glucose. We need ATP for that, a lot of ATP it turns out. So this is not showing you mechanism, it's just saying, look, we need ATP. And then when the liver makes glucose, if it's got a large amount of it, it can store it as glycogen. But then when it breaks glycogen down and it makes glucose, it will use only what it needs for its own metabolism, and then it will send the rest of the glucose out. And so there's a specific mechanism by which the liver sends the rest of the glucose out, and the liver has that mechanism and the muscle doesn't. Okay, so the muscle gets glucose, it doesn't want to send it out to somebody else. It is the tissue that's using it. It's doing the work. Right? Whereas liver is adapted through its enzyme systems, right? Through distinctions in the enzyme systems between muscle and liver. Which enzymes? Well, the ones we're studying that make liver do something different. Okay. So this cycle of glucose and lactate is called the Cori cycle. So here is something about the TCA cycle as a hub, and we're kind of skipping some aspects of this in the lecture that we had to skip because of the snow day. But I think we're kind of going to get the basic idea here. So here's the TCA cycle, and so blue going up is gluconeogenesis, right? It's the pathway that's basically mainly the reverse of glycolysis. If I slip and I say gluconeogenesis is the reverse of glycolysis, that's wrong. It's not the reverse. 7 of the 10 enzymes are, in fact, the reverse in the sense that they're the same enzyme and they're running backwards. And so enzymes can run in both directions, and which way they go depends on allosteric effectors and depends on the concentrations of substrate and product that are around. Right? Depends on that Q ratio, which you can also think of as a mass action kind of thing. So here's a TCA cycle, and we can have what are called glucogenic amino acids, which are amino acids that through their catabolic pathways, we won't talk about these pathways in detail, but we can just know that some of those amino acids can be catabolized to pyruvate or other citric acid cycle compounds. And if they can be, then they're in the cycle. And if they're in the cycle, then they're going to go through oxaloacetate, and if they're going through oxaloacetate, they have the capacity to go back through gluconeogenesis. And so you can note here that there are also ways for fats, triacylglycerols, the glycerol backbone of the fat, which is kind of left over after you're burning it. You're kind of burning the hydrocarbon parts, as we'll see. But then you got this backbone, don't want to let it go to waste. It can be fed back up into gluconeogenesis. And here are plants using light energy to react carbon in the air to put it into organic compounds in their soluble cell compartments. And it turns out that they make 3-phosphoglycerate, which is familiar as a glycolytic metabolite, so then it's going to go back as well. So these are just some of the possible ways to input into the TCA cycle. This isn't showing you outputs, but there are also outputs from the TCA cycle. Okay. So the compounds that feed gluconeogenesis, the 3 important compounds are lactate, pyruvate, and glycerol from fat breakdown. Okay. So it says here, occurs in the liver. It also, I don't mean to say it occurs only in the liver, so I probably should. It does occur in other tissues. It occurs in muscle, any tissue that wants to store, that wants to make glucose, and wants to store it as glycogen would have gluconeogenesis. But the distinction in liver is that when the glucose is made, it's then exported to get to the muscle. So this is just a list of glucogenic amino acids, and grouped by site of entry, so you can see it's actually most of them. And there are complicated catabolic pathways, as I mentioned. We're not going talk about them. But I do want to mention these transamination reactions. We can come back to them again.